My name is Engebretsen, and I am a medical doctor at the Department of Dermatology and Allergy at Herlev and Gentofte Hospital in Copenhagen, Denmark. I am the first author of this study where we have investigated the effect of season on some important skin barrier measures. We chose to do this study because in our research group we are interested in the effect of climate on skin barrier function and the risk of dermatitis. We often experience that patients complained of dry skin and eczema during the cold winter months in Denmark. And several studies have shown that low temperature and low humidity might increase the risk of dermatitis and have a negative effect on the skin barrier. Cheek and hand skin is heavily weather exposed, and interestingly, patients with atopic dermatitis often experience eczema in these anatomical locations. Furthermore, previous studies have shown that small protrusions on the skin cell surface is increased in patients with dermatitis, and also if there is a deficiency of natural moisturizing factors, which is derived from the important skin barrier protein called filaggrin. So in this study, we included 80 healthy participants, 40 male and 40 female, and they were equally divided into two age groups, 40 aged 40 years or below and 40 aged 70 years or above. And the participants, they were tape stripped on the cheek and on the dorsal aspect of the hand during the winter and the summer. And the tape strips, they were analyzed for natural moisturizing factors and also corneocyte surface structure. We also recorded if the participants had used any cream or moisturizers on the day of the sampling, if they had taken a shower or a bath. And during the winter, we also recorded the lowest temperature of the day. And during the summer, we asked the participants about recent sun exposure and use of sunscreen to estimate how much UV light they had been exposed to. And this was done to be able to correct for possible confounders in our statistical analysis. In this study, we found that NMF levels and protrusion on corneocytes, they varied significantly according to season. In cheek skin, the NMF levels were significantly lower and the number of protrusions higher in the winter compared to the summer. Furthermore, old participants had higher level of NMFs on cheek skin compared to young participants. Interestingly, high self-reported UV exposure during the summer was also associated to increased numbers of corneocyte surface protrusions. Finally, female participants compared to male participants had higher NMF levels on their hand during the winter and summer. This study is relevant as it demonstrates that important skin barrier measures are significantly affected by climate, both on an ultrastructural but also a biochemical level. Our findings add new pieces to the puzzle, and it might help explain why many experience dry skin and dermatitis when they get older, but also during the cold winter months. We believe that daily application of moisturizers could be an easy and feasible preventive measure to avoid this problem during the winter, but also when the patients get older.